So hi everyone, and welcome to the fourth edition of our MJS BioLinks Breathe and Learn. I'm Steve McKend. I work here in business development at BioLinks, and it is my pleasure today to introduce our guest. Craig Powell is joining us here from Vector Laboratories, and he's going to walk us through a simple process that will make a big impact on the clarity of your images. So we're going to dive right into how you can reduce unwanted autofluorescence in your tissue sections with Vector TrueView. So as usual, this will just be a quick 15-minute demonstration. And if you have any questions, you can put them in the chat below. And we've set aside 15 minutes afterwards to get to all those for you. And before we jump in, I was hoping we can make this a little more personal and change our Zoom names to our real names. So if you go to your tile, you can easily change your name from your Zoom name to your real one if you're comfortable. And while you all do that, I'm just going to run through the necessary Zoom housekeeping items. First thing is this session is being recorded, and we're going to send it to you right after the demonstration is finished. And out of courtesy to everyone on the call, I'm going to mute all of you, but afterwards, if you have any questions, feel free to unmute yourself and ask away. And with that, I'll pass it off to Craig, who's going to jump right in here.
Terrific. Thanks very much, Steve. And hello to everyone that's had the opportunity to join us today. As Steve mentioned, today's topic is on reducing autofluorescence in your tissue specimen. Now, autofluorescence is particularly problematic for many investigators working with immunofluorescence detection methodologies. The autofluorescence itself might be ubiquitous across the preparation, like a tissue section, and it may be masking the specific staining that you're trying to identify. Or indeed, it might be a little more subtle, such as the example I've got here on this particular image. For example, the green here is highlighting cytokeratin with a specific antibody. If indeed you're working on, say, a treated specimen or something that might be a disease state, you might think, wow, look at this cytokeratin infiltration into this dermal area, when in fact it's actually autofluorescence. This is an example of that autofluorescence that's now been quenched. So indeed, the more subtle autofluorescence could be interpreted as specific staining, be reported, and that would be a false result. So certainly this is problematic for many investigators. So let's have a quick look at what is autofluorescence for the 15 minutes I've got you here. What are some of the conventional or historical treatments that people have used to reduce or eliminate the autofluorescence in their preps? And I'll introduce you to our Vector TrueView autofluorescence quenching kits. And then as Steve mentioned, we'll have a bit of Q&A at the end. Okay, so what is autofluorescence? For those of you who have tuned in here and might be looking to get into immunofluorescence, certainly be aware that some tissue does have these fluorescent components. And the image here on the right-hand side, I think, demonstrates that. So the fluorescence I'm referring to here is inherent or endogenous to the tissue preparation, and certainly might be also due to aldehyde-based fixation. And that might be through paraformaldehyde, 10% neutral buffered formalin, any of those aldehyde-based fixatives. And you can certainly see from this particular image on the right-hand side here, some of these red blood cells, highly fluorescent, formalin fixation contributing here in this arterial wall, and the collagens and elastins here also being particularly problematic. These are the main sources of autofluorescence and that are particularly problematic for a lot of investigators. And you can imagine if you were working in the green spectrum, if you had a GFP protein, for example, green fluorescent protein, or you were using a standard, say, fluorescein or an Alexa 488 or a dialate 488, and trying to identify a marker here, you're going to have a lot of problems in trying to identify what is specific from that background autofluorescence. Let's have a quick look at some of the conventional treatments that have been used out there. Some are chemical treatments, and the ones that tend to be more widely used are the home brew methods, such as sodium borohydride or indeed potassium permanganate. They're pretty dreadful things to make up and also toxic. Sodium borohydride bubbles. Also, it's got a very short working life. I think about 30 to 60 minutes, and then it won't be effective. And certainly when you do apply it, as I'll show you in a subsequent slide here, it's really not effective against red blood cells. Other treatments that investigators have tried have been using UV light photo treatments to quench or eliminate this background autofluorescence. And it's not practical for using large volumes of slides. You have to put each individual slide under that UV light of, say, 360, 365 nanometers to quench that fluorescence. And the thing is, it also does take quite a period of time. It takes up to about 24 hours to quench that. So you can certainly see that that is not very practical for a lot of investigators. Other treatments are masking treatments, some of these ink-based reagents. I will say that the ink-based reagents are effective against something called lipofuscin, which is another source of autofluorescence. You tend to see that more in aged mammalian brain. But I think most of investigators listening in here would have problems with those reagents that I meant, with those inherent cellular components I mentioned earlier on, the red blood cells, your collagens, elastins, and induced through the use of aldehyde fixation. So those ink-based reagents aren't really effective against those cellular reagents or indeed against the use of an aldehyde-induced fixation autofluorescence. Other quenching methods that investigators have used has been a copper sulfate-based solution. A couple of things with the copper sulfate, though, is when you do apply it, while you do get a quenching effect, you're going to wash off the excess material so you can then cover slip it or whatever you want to do, maybe counterstain it. And when you do that, the copper is removed and therefore all the autofluorescence pops back up. So it has to actually be present for it to be effective. And as I'll show you in this next slide, these reagents really don't give you a nice, deep, dark, black background in which to then visualize your specific signal. So going back to my original image there, showing you the problems with the inherent autofluorescence due to, say, red blood cells, collagens, elastins, or indeed induced through the use of an aldehyde-based fixative. Copper sulfate, you can still see some of the collagen in there, the aldehyde-fixed residual fluorescence, and the red blood cells. This is an ink-based reagent, Sudan black. Again, not particularly effective against some of these compounds or cellular components. And the sodium borohydride, dreadful to make up, as I mentioned, and really does nothing to quench the autofluorescence from these red blood cells. All right, so some of you may have tried those and experienced some of these phenomena as well. They're not really effective, even though they have been published on by some investigators. So let me introduce you to something called our Vector TrueView autofluorescence quenching reagent. As I've listed there, it's a novel technology, solution based on hydrophilic molecules that bind to the tissue and eliminate and greatly reduce also background autofluorescence. Now, I will say we actually do refer to it as an electrostatic binding of the reagent to the tissue. It is reversible, so if that's something you do want to remove, you can then let it wash in buffer for an extended period and it will remove it, and then you can reapply it to quench it. So there is that little trick you can use if you want to go back, see what the original autofluorescence was, make sure it's not affecting anything you're trying to interpret. But nonetheless, the TrueView itself is an aqueous-based, non-fluorescent solution. Very short treatment of about 2 to 5 minutes. You give a quick PBS wash afterwards, and then you can cover slip with the anti-fade mounting media. And it's very easy to scale up for a multiple number of slides. You can put it in a Copeland jar or a staining rack, something like that, and move the slides in and out quite easily. So again, going back to this original image I showed you, it's been problematic with the red blood cells, these other cellular components, and the autofluorescence due to aldehyde fixation. Here's the copper sulfate, a quenching reagent's been used, the ink-based reagent, and the sodium borohydride. This, believe it or not, is actually an adjacent section of human pancreas treated with TrueView. So it's one of these sections that does not have any specific staining, such as these, but treated with TrueView. And you get an appreciation of the drop of that autofluorescence when treated with TrueView. Now, some of you might be working with established SOPs that taking a long time to work up and optimize. This is just to give you a bit of an idea of what I'd refer to as a fairly routine or standard immunofluorescence detection methodology. You're doing your standard blocking, primary antibody incubation. Obviously, you do a wash. Then I put on your conjugated secondary antibody with a fluorophore, your choice, whatever it might be you're working with, an Alexa, a Dialyte, or say a Texas Red. Then you put on the TrueView at this step after you've applied your fluorophore for that short period of time, give it that brief rinse, cover slip, and then visualize. So the idea of this slide is to show you there's no great optimization required. You simply slip the TrueView in after you've done all the upstream detection, and then you do the cover slip. This is a bit of a schematic on showing you what I'm referring to. Here would be your target antigen, that white dot, detected with a primary antibody, and then subsequently with this fluorophore conjugated secondary antibody. And you may be experiencing this endogenous autofluorescence. After the detection methodology, you treat with TrueView, and the background goes away, this autofluorescence, and retains the specific signal. So hopefully you can see my hands, I'm waving around here. You've got the specific signal and background autofluorescence. TrueView retains that specific signal, but drops down that background autofluorescence from those cellular components I mentioned, and through and from the use of an aldehyde-based fixative. Now, importantly, I will say that TrueView does turn sections blue, and I'll show you a quick little video coming up. But nonetheless, autofluorescence itself is a form of background. So it's important to determine if you are experiencing background in your immunofluorescence. Is it due to autofluorescence, or indeed has it been introduced from some part of the detection system? Now, in the time we've got here, I'm not going to get into that specifically, but nonetheless, you can easily determine autofluorescence in your prep by cutting a section, not treating it with any detection reagents, simply looking at it under the microscope, and are there fluorescent compounds, reagents, cellular components on that slide that determines whether or not any autofluorescence is present and not due to the detection reagents. All right, so let's put this into a little bit more of a practical situation, and how is actually TrueView applied? All right, a very easy methodology. Reagent A, B, and C, mix them together in that order, and simply apply it to the tissue section for your 2 to 5 minutes. Now, some of you might be thinking out there, okay, but I work in a defined channel, a green or what have you. How does it actually work on some problematic tissues as well under these different spectra? And here's human kidney sections, some serial sections here. And this particular section is simply just a cut tissue section of human kidney looked at under the green channel. I will say that it has been counterstained with DAPI, so we can identify some of the underlying tissue morphology and architecture. And this is the extent of autofluorescence in the green channel. So when we treat it with TrueView, what happens? All of that background autofluorescence is eliminated, and you can certainly see now the nice blue DAPI nuclear counterstain and a deep dark background. So if you did have a specific green signal there you were trying to identify, this would make it certainly a lot easier. Here it is in the red channel, again, human kidney, and counterstained just with DAPI, no other detection reagents in there. You know, the red marks that are in here is all autofluorescence. So what happens under the red channel when you treat with TrueView? Again, the autofluorescence is eliminated, and the retention here of the DAPI provides a bit of an idea about that underlying morphology, and certainly then would allow you to clearly identify your red signal. Another particularly problematic tissue that some investigators work with is human spleen or spleen tissue. And this particular image here does not have any treatment for reducing autofluorescence, but it is a double stain. So this gives you an idea of the retention of that specific signal, but the drop in the background when you treat with TrueView on this next slide. But I think it's important to show these side by side. So this is actually double stained. It's CD20 in the red, which hopefully you can see, reddy orange, and then Ki67, which actually you can't see in here. They're like little pinpoints hidden in here. It's a mitotic marker, Ki67 or Ki67. You can't see them without reducing the autofluorescence. So what does this particular section look like when, as a double label, when treated with TrueView? And these white arrows here certainly show you these green dots, the starry sky, one of our colleagues mentioned, is actually true staining for Ki67. And the other arrow here showing you this red staining for CD20. So if you look at these, compare them, obviously it would not be practical to identify Ki67 in this untreated tissue. Okay, and certainly in the red channel here, again, specific staining for a cytokeratin marker, and this is counterstained with a DAPI. These red blood cells could be interfering with what you're trying to interpret here. This is just an example. When you treat with TrueView, all of the red blood cells, autofluorescence has been eliminated. All right, hopefully that gives you a bit of a picture about the action of TrueView. It actually comes in a couple of different catalog numbers. One is SP8400, which is just referred to as TrueView Autofluorescence Quenching Kit. And the other one is SP8500, which is referred to as TrueView Autofluorescence Quenching Kit with DAPI. And the point I've made here is that both of these kits are supplied with Vector Shield Vibrance Anti-Fade Mounting Media. One without DAPI, that's the 8400, and one with the counterstain DAPI mixed in, the 8500. And for those of you who aren't familiar with perhaps that methodology of counterstaining, particularly maybe with TrueView or an anti-fade reagent, got another quick video for you here. So just to let you know, Vector Shield Vibrance is one of our newer formulations of Vector Shield. It is a curing or a setting media that allows you to simply cover slip and avoid having to put, say, on nail polish or a sealant around the outside of the cover slip. All right, I'd like to say thank you very much for your time, and I'll open the floor up to any questions you may have. Thank you.
Thanks, Craig. If you have any questions, you can pop them in the chat below. I don't see any in there right now. Oh, here we go. Can you read that, Craig, in the chat?
Yeah, let me just open the box up here a little bit. Under what conditions would TrueView fail? Is TrueView meant to eliminate autofluorescence versus a background noise? Work with rat vessels, fixed with PFA, and TrueView does not seem to work at reducing or eliminating background signal. I think it's important to identify whether or not the autofluorescence you've got there, or the fluorescence you've got there, is indeed due to autofluorescence, as I mentioned earlier on, or due to background signal. Simply cut a tissue section, look at it under the spectrum you're working on. Is it green, red, far red, something like that? If you do have signal, then that is due to autofluorescence, obviously. But if there's no signal there, more than likely it's due to something that's been introduced, a detection reagent or something like that. The other part that I haven't really touched on is, and I don't think it's in this, is lipofuscin, which is more of a punctate sort of autofluorescence. It's not the overall toning you might see with the autofluorescence I was referring to earlier on. So I would highly recommend running some defined negative controls in your assay. TrueView will reduce, usually eliminate, autofluorescence, as I mentioned, from red blood cells, collagens, elastins, or due to the use of an aldehyde fixative. 2 to 5 minutes is worked up on a thin cut sections of 4 to 6 microns. If you're working on a thicker section, maybe on the rat vessels, you're working on a 20 or something like that, leave it longer. How long? Leave it another 5 minutes. It's not going to hurt anything, and see if that will penetrate in. If indeed you do see autofluorescence due to the collagens or elastins in there. So just leave it a little longer, give it a chance to work. You might need to permeabilize those vessels as well with a detergent and give it a chance to act. Okay, hopefully that answered that particular question. Do you still add TrueView after secondary antibody if your counter stain is separate from your cover slipping agent? Or would you add TrueView after counter stain in this instance? You know, your choice. It doesn't really matter. As I showed earlier on, you could certainly apply TrueView between putting on your conjugated fluorophore and the counter stain, or you could put it on at the end. TrueView won't affect the DAPI counter stain. So certainly give it a try, see what's more convenient at your end in terms of your given workflow. But we do recommend, in most cases, applying TrueView after your detection reagents because it can be eluted off. As I mentioned earlier on, some extensive washing could wash off the effect of TrueView and pop up that autofluorescence background. So that's generally why we recommend applying TrueView after your detection reagents. Can you use with whole tissue like lung before sectioning? So again, I'm going to interpret that as being, before you actually section it, would it be effective? And the answer is probably no, because as I mentioned, it is a reversible binding interaction. And once you go through the process, possibly fixing, washing, etc., incubations, there may be problems with that TrueView being washed off. So it is intended to be applied at the end of the immunofluorescence workflow. But certainly on something like lung, it would be highly effective. I've had investigators working on, some very dense collagenous type tissue. And what they've done is actually leave it longer, as I mentioned in one of the earlier questions, and it was effective in reducing that autofluorescence. So I've got a question here. I'm just reading through it. We've sometimes used glycine added to our wash buffers to reduce autofluorescence from fixation. I assume you think that TrueView will work better than this for such purposes. Does the TrueView work in fixed as well as frozen tissue section? So I'll answer that first. Yes. Certainly, we've evaluated it both on FFPE, formalin fixed paraffin embedded, as well as frozen tissue sections that might have been fixed with PFA, an aldehyde fixative, or indeed with an acetone fixative as well that might have highly fluorescent cellular components. So yes, it certainly will be effective in that. Is it more effective than glycine? You know what? I don't know that I've actually got an answer for you on that in terms of have we run it in side by side. I'd have to say that, if the glycine is working for you, then that is great. But I'd have to say that if indeed you have persistent, residual, or underlying toning autofluorescence due to aldehyde fixation or some of those other cellular components, the TruVu will get rid of it completely. So just keep that in mind as you're working through your application. Do you have any info on TruVu with an organoid section? Sort of touched on that a little bit with the thin cut sections versus the thicker sections, or indeed a whole mount preparation. I did have an investigator working with 200 micron sections. They sent me a nice 3D image of it. It was really pretty. And they used TruVu on that. Basically, again, they just left it longer than the 2 to 5 minutes that we've given as a time frame for the thin cut section. So on something like an organoid, yes, certainly if the detection reagents have access to the inner part, I'm thinking a brain or whatever you've got there, or CNS type tissue as an organoid, and you poke some holes in that with a detergent or something like that. If those detection reagents can get in, then the TruVu can get in too. So just leave it a little longer. Don't necessarily wash the living daylights out of it after you've applied the TruVu, but certainly give it a decent wash. Get rid of the excess TruVu, and then you could certainly visualize that. We sometimes get autofluorescence from matrigel residue. Yeah, certainly I'd recommend trying to get rid of that residue. I don't know what's in matrigel and whether or not TruVu would reduce that. If it does, let me know. that'd be great. That way we got a little bit of collaboration going, and certainly work with the team there at MJS Biolinks to supply that data. And that'd be great. We can then put that in our database and help other investigators too. We've also seen some autofluorescence in frozen sections that were first treated with sucrose immersion prior to fixation. Yeah, so sucrose is used as a cryoprotective. I actually used that on primate embryos once upon a time to retain that morphology. This is on post-implantation embryos of, I'm trying to think what stage they were, maybe stage 12 through 15 or 16 or something like that. Yeah, I had some issues with that as well. Wash the sucrose out if you can do that. If that's not working for you, try the TruVu. I don't have data on that at this time. Have a look and see whether it isn't a specific spectrum. Is it just in the green or the red, or can you account for it in certain cell types as well that might have taken up the sucrose? Give it a thorough wash, as I mentioned, prior to or post fixation. I don't know how thick the preparation is. Obviously, the thicker the prep, the longer you're going to have to wash, and the more washes you're going to have to use as well. And you might have to try a series of treatments, but try the TruVu and see how that works. Can TruVu be used in conjunction with Sudan Black for lipofuscin-rich tissues? Yes, it can. If you'd like a procedure on that, drop a quick note to the tech team at MJS Biolinks, and we can provide you with that. We did have a colleague in the UK who did that, and they worked up a bit of a protocol and gave it to us. So I'm more than happy to share that info and supply it with you. If you see autofluorescence from both sources, maybe you're working on a neurodegenerative type model, an Alzheimer's, Parkinson's type disease model, where you do see both lipofuscin and possibly autofluorescence from some of these other cellular components, etc. So yeah, certainly ask the tech team for that. And certainly anything to do with pricing or samples, I'll pass off to MJS Biolinks to accommodate those requests.
Craig, there was a question about using TrueView on cell monolayers. Is that possible?
Yeah, so I imagine that the monolayer itself is probably fixed, either with probably an aldehyde-based fixative, a PFA, maybe a 2 to 4%. Yes, certainly it's not that the pH will be problematic in that instance, or you know, cause cells to either crenate or pop open with, you know, problems with osmolarity or tonicity or something. No, that it should be fine to use on those monolayers. Yes.
Great.
Thank you.
Well, if there are no more questions, thanks so much, Craig. I think I could listen to you talk about that all day. But that will conclude our session for today. And I want to thank all you guys for coming. I really hope you enjoyed it. And as Craig said, if you have any more questions, sample requests, quotes, or just want to give us a call to chat, feel free to email us or call us using the information on the slide right there. And keep an eye out for your Timmy's gift card. Your coffee is on the way.
Thank you, Craig. Thank you, Steve.
You're welcome. Thank you all very much. Have a good day.
Thanks, Craig.